Hello everyone, and welcome. Today, we'll be diving into the fascinating world of neuroactive steroids and peptides. As you can see, sleep stages are important. I'm MS Nadeem from KAU Jeddah. Let's explore how these molecules impact our brains and well-being. Let's start with neuroactive steroids. These are steroids that can rapidly alter neuronal excitability. The term neurosteroids was coined in 1992. They're produced in the brain or by endocrine glands and have a wide range of clinical applications. Neurosteroids can be classified into three types based on their activity: inhibitory, excitatory, and pheromones. Today, we'll focus on inhibitory neurosteroids, which act as positive modulators of GABA receptors, exhibiting antidepressant and anxiolytic effects. Here are some examples of neurosteroids, including pregnans, gonane, and estrane. Steroid structures consist of four fused rings, named A, B, C, and D, known as a gonane core. Steroids are named as derivatives of this core. This slide illustrates the basic structures of androstane, pregnane, and colestane. Notice the differences in their carbon counts and side chains. These structural variations contribute to their diverse biological activities. Pregnanes are steroid derivatives with 21 carbons and are the parent structure of progesterone. Pregnane is a parent hydrocarbon for two series of steroids, 5A, pregnan, and 5B, pregnan. Progesterone is a crucial hormone derived from pregnan. Let's look at some specific examples. Pregnenolone enhances myelinization and improves cognitive function. 5A, dihydroprogesterone and dihydrodeoxycorticosterone are agonists of the progesterone receptor and positive allosteric modulators of the GABA receptor. Progesterone, formed from cholesterol in the brain, promotes myelination and inhibits the neuronal nicotinic acetylcholine receptor. It's also a precursor to pregnenolone, which is synthesized by Schwann cells. Androstanes, like androstenol and androsterone, are C19 steroidal hydrocarbons. Androstenol reduces anxiety, while androsterone is an inhibitory neurosteroid that acts as a positive allosteric modulator of the GABA receptor. Cholesterol is essential for neuronal physiology, regulating ion permeability and transmembrane signaling. It's a crucial building block for many neurosteroids and plays a vital role in brain function. Excitatory neurosteroids have the opposite effect, acting as negative allosteric modulators of GABA receptors and weak modulators of NMDA receptors. They can have antidepressant and anxiogenic effects. Here are some examples of 3B hydroxysteroids, including pregnenolone sulfate, PS, dehydroepiandrosterone, DHEA, and dehydroepiandrosterone sulfate, DHEAS. We also have pregnans like isopreganolone and epipregnanolone. Now let's briefly touch on pheromones. These are chemical signals used for communication between individuals, particularly in insects. Slide 13 lacks detailed content. Neurosteroids play a crucial role in sexually dimorphic behaviors and emotional responses. Their levels increase during acute stress and are associated with changes in mood, sexual desire, and anxiety. Alterations in neurosteroid levels are linked to women's mental health conditions, including premenstrual syndrome, PMS, postpartum depression, PPD, catamenial epilepsy, CE, and postpartum psychosis, PPS. Synthetic neurosteroids like ganaxolone have been investigated for use against epilepsy. Ganaxolone, an analog of progesterone, acts as a positive modulator of GABA receptors. 4. 16-androstadione. 3B. OWOL is a synthetic pheromone under investigation for anxiety disorder in women. Androsterone is a putative human pheromone that may make men sexually appealing to women. Now let's shift our focus to neuroactive peptides, also known as neuropeptides. These molecules play a critical role in neuronal communication. Slide 18 lacks detailed content. Neuropeptides are peptides that affect communication between neurons, acting as neurotransmitters or neuromodulators. There are approximately 100 different neuropeptides in the brain. A neuropeptide is a small-sized protein molecule produced and secreted by the cells of the nervous system, playing a specific role in the regulation of the nervous system. Unlike neurotransmitters, neuropeptides are not recycled after secretion. They often undergo post-translational modifications and are coded by at least 70 genes in the mammalian genome. Opioid neuropeptides are produced endogenously and act as modulators of neurotransmitters in the CNS. They alter the electrical properties of target neurons, acting as pain relievers and regulating behaviors like euphoria. Endorphins are produced in the pituitary glands and hypothalamus in response to stress or pain. They inhibit pain perception, affect mood, and cause euphoria. There are at least 20 different endorphins in the human body. So how can we boost our endorphin levels? Activities like meditation, laughter, acupuncture, running, eating spicy food, and listening to music can all trigger the release of endorphins. Endorphin receptors include mu, kappa, K, and delta receptors. Mu receptors are the target for morphine and endorphins alpha and beta. Kappa receptors have high affinity for ketocyclosacine and dynorphin. Enkephalins are naturally produced in the brain and spinal cord to stop pain messages. They promote a sense of well-being and reduce pain by increasing the release of acetylcholine. Dynorphins regulate the electrical activity of neurons, inhibit oxytocin secretion, control appetite, and modulate the pain response. Dynorphin A and dynorphin B have high binding affinity with the kappa opioid receptor. Endomorphins are endogenous opioid peptides with the highest known affinity and specificity for the mu opioid receptor. They are released during pleasurable activities like exercise, massage, eating, and sex. Opioid receptors activate a GP protein, which inhibits AC, decreasing CAMP levels. This leads to decreased Ca++ conductance and increased K+ conductance, resulting in hyperpolarization and decreased neuronal excitability, ultimately relieving pain. Now let's move on to some other important neuropeptides. Slide 30 lacks detailed content. Corticotropin releasing factor, CRF, is a hypothalamic peptide responsible for orchestrating endocrine, immune, and behavioral responses to stress. Peripherally administered CRF does not cross the blood-brain barrier. CRF neurons are distributed in the hypothalamus and the central nucleus of the amygdala. This distribution highlights its role in stress response and emotional regulation. This diagram illustrates the hypothalamic pituitary adrenal, HPA, axis. Stresses trigger the release of CRF, which stimulates the pituitary gland to release ACTH, leading to cortisol production and stress relief. CRF binds to GPCRs, activating Gs or GI. Gs activates adenylate cyclicy, converting ATP into CAMP, which activates pKa. pKa activates CREB, increasing the gene expression of pro-opiomelanocortin, POMC. This slide simplifies the CRF mechanism of action, showing the cascade from CRF binding to GPCRs to the release of ACTH into the blood circulation. Orexin, also known as hypocretin, is a neuropeptide produced in the hypothalamus, regulating energy homeostasis, sleep, and appetite. There are two types, orexin A and orexin B. Orexin A binds to OX1R receptors, while orexin B binds to OX2R receptors. These receptors are coupled to different G proteins, leading to diverse downstream effects. This diagram illustrates the sleep phases, highlighting the importance of REM sleep. Orexin neurons are most active during wakefulness and least active during non-REM and REM sleep. Deep sleep and REM sleep are crucial for restorative sleep, leading to better health and performance. Deep sleep is associated with growth and healing, while REM sleep is important for memory. Orexin signaling leads to the elevation of K2+ and activation of the NAR-CAR exchanger, resulting in the depolarization of the target neuron. It also inhibits the GERK channel and increases NMDA receptor expression. Sleep aids often work by acting as orexin receptor antagonists. These substances counter the signaling effects of orexins, promoting sleep by reducing wakefulness and alertness. This table summarizes the development status of several dual orexin receptor antagonist DORA compounds, such as suvorexant, which is used to treat insomnia. Melanin-concentrating hormone, MCH, is a cyclic 19 amino acid orexigenic hypothalamic peptide, originally isolated from the pituitary gland of teleost fish. MCH binds to MCHR1 and MCHR2 receptors, reduces the effect of glutamate, and is implicated in energy homeostasis, wakefulness, sleep, motor activity, and motivation. Somatostatin is a mixture of two peptides produced by the ventral hypothalamus. It inhibits the release of growth hormone, GH, and thyroid-stimulating hormone, TSH, from the pituitary gland. High concentrations of somatostatin can reduce or inhibit the production of many endocrine hormones, such as insulin, potentially leading to diabetes. Thank you for your attention. Now, I'd be happy to answer any questions you may have and open the floor for discussion.